All right, thank you, and thank you to the organizers for inviting me. So today I'm going to talk about a few different sort of stories. Just so you know, I'm a computer scientist. I work at the intersection of deep learning, machine learning, as well as applied math, and I generally try to develop new types of methods for biomedical and increasingly neuroscientific problems. So something that's catalyzing more of my entry into neuroscience is the new WOOTSI Institute we have at Yale. When I think, so that's kind of the perspective that I'm coming from. So when I think about sort of connectomics in neuroscience, what I think of is kind of building sort of a parts list in a way and seeing how they all work together, in a vague way, like a computer system. So one of the projects that I'll talk to you about involved kind of learning the architecture having to do with the 181 neural pill or nerve ring neurons and their connectivity together with a collaborator, Danielle Colon-Ramos at Yale. And first I'll kind of deviate and talk a little bit about the methods and kind of the philosophy behind a lot of my methods. And then I'll return to applying it to this question. One of the interesting things about biological circuits of any kind, but neuronal circuits, is that they vary in scale and structure. So towards the end of that topic, I'll talk about worms at different stages of development. These are all C. elegans worms. And you can think about them at the cell level, at groups of cells, which we call adjacencies, because we turn them into a graph, circuits, systems. And one of the frameworks that I'll show is you can think of these using topological data analysis, which is a natural fit for the multiple scales. But we tend to combine topological data analysis also with a notion of what is the geometry of this space where this analysis is taking place. So that's where the geometry comes from. And then finally, I'll show when you want to learn or infer stuff, you can add deep learning to the mix, and that helps you. So in general, some of the challenges that we have in neuroscientific systems, as we do in other biological systems, is how do you go from high-dimensional neural activations or connectivities to really, interpretable, understandable insights? And confounding a lot of that is a lot of noise in most of our kinds of neuroscience data, and distributed representations of these within the brain, also within the connectivity. And so the other interesting ingredient is the time variance that we have. And I'll get to that in the second part of the story, where I'll actually try to use the activity dynamics to infer a functional connectivity rather than a structural connectivity. I do want to mention that I will be giving some examples from cellular data, because actually I've worked a lot on cellular level high-dimensional data, and this can include single-cell RNA sequencing data. I initially started working with high-dimensional mass cytometry data when I first entered biology. And you have similar challenges there. You have very noisy data, many parts, different connectivities. And so when I show my methods, I'll tend to show an example, sometimes on cellular data, and then show how I applied it to neuroscientific data. And a lot of the analysis tasks that we've worked on developing methods for in our lab, they, in a sense, tend to be unsupervised or self-supervised, at least. So we've worked a lot on denoising techniques, visualization of these high-dimensional entities into low-dimensional representations that are hopefully more understandable, where you can understand kind of the structure and the shape of the data, if you will. Inferring what's really generating these behaviors and entities in terms of the underlying regulatory network and dynamics from static snapshot data, which is actually better in neuroscience than in other fields. And as I mentioned, these are the general range of the techniques that I'm sort of using. So the first technique I'll talk about will have to do with this multi-scale organization that we were trying to discover, given this neuroscientific data that came from the C. elegans worms. Initially, when my collaborator actually gave me this data, I was like, oh, this is small data. I want to work on big data. But it turned out the data, in a way, is actually pretty big, and it was a very good method for actually developing this series of methods. So we first started developing this method and published in IEEE Big Data 2019. And then subsequently, we applied it to cellular and neuroscientific data in 2021 and 23. And then actually, together with Mason, we published this, I think you were our editor, in SciMods. He didn't give us...
Slightly, maybe.
Slightly, maybe. Okay. And so in 2023, kind of studying the properties of this method. Okay, so again, the motivation is these kinds of biological entities, when you look at their organization, they have multiple scales of organization and a high degree of complexity. So the natural thought for someone who's oriented with some kind of applied math, pure math, is to think about topological data analysis. And of course, that's based on algebraic topology, where you're trying to quantify aspects of the shape that are a little invariant to the scale. And these can include things like these Betty numbers that probably a lot of you already know about. Betty zeros, connected component, cycles, cavities. And so you have this idea that this ball here has one cavity and is all just one connected component, whereas the donut actually has two cycles, so it can be distinguished just using these numbers. And the thing about distilling things into numbers that's actually very appealing for someone oriented with machine learning is it's very easy to feed machine learning numbers. In a way, it's very hard to feed it these kinds of abstract shapes. So just to remind you of how very simple computational homology works, this is Vietoris Rips homology. And there usually you have these data in their multiple coordinate system. And you're looking at distances between them. And this could be something like a Euclidean distance. And when you apply computational homology, you grow an epsilon ball around these data points. And when they connect, you have the depth of these individual components, and you could have the birth of a new component. And you can trace this, and this is called zeroth order homology. You can also have first order homology, where you look at loops. And these loops can then be characterized, they're characterized in a different color, and when they appear and when they disappear is shown here. And of course, if there's a feature that's persistent, it lasts a little while. So there's a gap between the birth and the death. So it's not right on the diagonal. So that's very nice and interesting, but it actually hasn't worked for us that well in biological data, starting with cellular data. And some of the reasons that we were surmising is that there's a lot of inference going on from very noisy images, segmentations, or even noisier data like the single cell data that I talked about. And some of the pre-processing steps, whether it's segmentation or pre-processing, can actually add a little bit of inaccuracy to this. And so one of the solutions that we started developing early on, since maybe the start of my lab in about 2015, is to use the lower dimensional intrinsic space and redundancy in the graphs, whether it's graphs created from cells or neurons, to try to address this. And this is sort of called the manifold assumption, and it actually works unusually well in high-dimensional biological data. So the assumption is that when you have high-dimensional data, it's coming from some kind of lower dimensional entity that has shape, and actually you could roughly figure out the shape if you go from neighbor to neighbor. And how do you figure out what the neighbors are? Very similarly, you can, like a manifold, if you have closely sampled data points, you can use Euclidean distance locally to find who the neighbors are. And then globally, you take a walk along these data points, and that can give you an idea of the distance. So usually we have some noise inherent in these, and there's also this idea that you want to denoise it. And you might be asking, why is this a good assumption for scientific data in particular? It's because a lot of the features and even the connectivity features we have are highly constrained. They're constrained by biological, biophysical constraints, and there is a lot of sort of redundancy. So if one neuron is close to and connecting to another neuron, it can't be at the same time all the way around the worm in its tail if it's connecting in its head. So there's constraints that are happening. So what we do instead is we try to take this into account when we build graphs of entity. So instead of just assuming the ambient space has a totally trustworthy distance, we go for a graph that's based more on the manifold distances. So the way we do this, and again, I'm giving you an example of cells by genes, and I'll show it to you in neurons in a bit. So if you have your data, it can be high dimensional, it could also be low dimensional, it doesn't really matter. You can change from here to a distance. And from distances, we want to localize the distances because we don't believe the global Euclidean distances. So we localize them using affinities. And you see the minute that's localized, if I had a square affinity here, like a uniform kernel, this would be a nearest neighbors graph. And this really just follows the shape of the data. And you can already see it visually. Something we do to...
In the affinities?
Yeah.
Are you doing any sort of thresholding or anything, or you're just...
I usually don't mathematically threshold, but this visualization has a thresholding.
That's where it comes in. Okay.
Yeah, mathematically it's exactly this, but yeah. Yeah.
There's some kind of relationship between like using this Gaussian kernel affinity transformation and like assuming that the data are on a manifold.
What's that?
You motivated this as by thinking from a manifold perspective.
Yeah, yeah. So first off, remember that in a manifold, you can sort of discover, intuitively you can discover the shape of the manifold by following paths to near neighbors. So you're kind of staying close to the manifold. So this is the first step in doing that, is saying, okay, I'm really going to emphasize the connections to my near neighbors and de-emphasize the connection to my distal neighbors. And I'll show you in a second how that helps. Okay, so once you do that, once you compute the affinity matrix, something else you can do, and I've been told by certain kinds of mathematicians that this is outdated, but I use it all the time in very modern papers and it works, is transitioning this, so changing from this into a transition operator. And that I do simply by row normalizing these matrices. And so now I have a transition operator. The transition operator gives me the probability of jumping from any data point to another data point with a probability that's proportional to its affinity. That's basically what it gives. So now you can do literally what I said to discover the manifold, right? You can hop to your near neighbors with some probability and to their neighbors and their neighbors. And if you compute t-step probabilities on that, then you have your manifold distances, basically. Because those are paths along the manifold, because they're paths through the data. So that's kind of the connection. And that's why we tend to do this. That's why we tend to actually use these transition probabilities. And it also turns out, if you look at it spectrally, it denoises the spectrum of this graph that's been created. And so again, you have to raise this operator to the power of t. This is a Markov matrix, so it gives you t-step random walk probabilities. And this is a random walk matrix. You could also do this if you just started with an adjacency or affinity, which we often have in neuroscience, and then you still get a transition operator. You might recall the diffusion maps embedding. The diffusion maps embedding is an embedding into Euclidean space of these. So you have new coordinates that respect t-step diffusion distances. Now, we often don't use the actual map construction because it has some properties that may not be suitable, but we often use the operator. So I'll often talk about how to go from the operator to the next step that we use. In the maps construct, you simply eigendecompose the diffusion operator. You weight the vectors by the eigenvalues. You discard the 0th eigenvalue vector. And then the coordinates of your new points are just found in these indices.
I've always been confused about this. What is the difference between this idea and simply multidimensional scaling, in which I give you a matrix of distances? In this case, it's a diffusion at distances, and I just simply ask to map into an Euclidean space so that Euclidean distances match as well as possible.
This is a really fantastic question, and I'm going to get to that a little bit later. Because actually, we have a technique called fate that almost does exactly that. Instead of that.
Scaling is a technique that has been around like for tens of years.
So I'll tell you the difference between this and multidimensional scaling, and then I'll tell you how you can actually use multidimensional scaling if you suspend your disbelief for a few slides. The first thing is this is not the same as multidimensional scaling because it took the transition operator here It's eigendecomposing the transition operator. It doesn't actually run any kind of distance preservation, classical multidimensional scaling method, because it's really trying to preserve these so-called diffusion distances. So now the next question you might ask me is, what if I just computed these diffusion distances and then fed it to MDS? That's what I'll relate to FATE, my method, a little bit later on. It gives different results than this, and I'll show you why.
I'm curious, what do you put on the diagonal for this matrix? Is it ones?
It's ones.
Okay.
Usually, it depends on your kernel, but it's usually ones. Okay, and so anyway, this is an example of a coordinate system that uses this distance inherently, and I just wanted to bring it up. One thing that's been noted about these is if you use these kinds of coordinates like diffusion map or even Laplacian eigenmap, and the difference between this and Laplacian eigenmaps is that Laplacian eigenmaps don't have this powering, which turns out to be important for denoising. You can get clusters that follow the data manifold as opposed to other methods that don't. So we decided to create a topological filtration using this kind of graph instead of the original graph that we have. And this we've called diffusion filtration or diffusion condensation. And the idea here is we actually compute this Markov normalized affinity matrix, and then we just apply it to the data. You might wonder what happens when you apply it to the data. It replaces every data point with an average of its affinity neighbors. And so instead of giving you K clusters, because I don't know what K is, because that assumes I know the level at which the interesting organization in biology exists, I just sweep through all the scales. And this is what this process looks like. As I reapply and recompute the diffusion operator, you start to see different scales of this. And we've tested this on a lot of data sets, including very curvy data sets that line contorted spaces. And if you raise your T to a high enough power, it does a much better job than Vietoris-Rips of giving you multi-scale organization. This data set we created from 3 Gaussians, and you'll see at some point you get to 3. But you also see further groupings that might be interesting. You might be wondering what happens at the end of this process. At the end of this process, we are guaranteed to go down to 1 point. And I'll talk to you a little bit about how we can prove that. If you want to think about this stuff spectrally, which is sometimes important, you can think of this graph of points and the original features or whatever coordinate features you have as being features on this graph. And what you're actually doing implicitly when you apply the diffusion operator is something like you're doing a graph Fourier transform, which is looking at the eigenvectors of either the graph Laplacian, which normalized in a certain way is the same eigenvectors as the diffusion operator. And then you're performing a low-pass filter on these. So if you raise T to a low power, you get a little bit of filtration. If you raise T to a high power, you get more filtration. So you're cutting off the high-frequency parts of this spectrum, which can be useful if you have very noisy data or if you have noisy connections like we have. If you want to think about it again spatially, it is giving you spatial smoothing, which is the classic signal processing thing. The low-pass filtering is spatial smoothing. We originally used this process just to denoise the data, only one iteration of it. This was a method called MAGIC that came out in Cell. When single-cell RNA sequencing first came out, you couldn't see any structure in the data. And this is a worm data set along the course of development. We simulated what's true about single-cell data, which is that it's about 80% zeros. Just take 80% of it, just turn it all to zeros. And then we applied one iteration of this step, and you see you re-impute the original trend back, but it's even less noisy than the original trend. So that's kind of what it was. But here we apply it over and over again to give us multiple scales of organization. It's like you're killing variation each time to get you multiple scales. So the way we can actually show that these points all converge to 1 is you have, at each point, you can think of the convex hull of your data points. But because you're replacing every data point by its weighted average of its neighbors, it'll shrink the convex hull. And so you'll get a series of nested convex hulls that actually go. And the shrinking rate is proportional to the smallest value in your kernel. So if you have a Gaussian kernel, it's proportional to the smallest. There is also a much longer spectral proof when you can look at it spectrally, and it's based on the first non-trivial eigenvalue. The first eigenvalue is always 1 in a Markov matrix, but this first non-trivial one, that's the one that's less than 1, dependent on that gap. So what do you get at the end of this process? So at the end of this process, you do get a 0th order homology using the condensation. So you can see on these different shapes what the persistence barcode looks like, as well as at each iteration, you can do higher order Vietoris Rips homology to characterize these things as well. And before I show you what the neuronal data looks like, I will talk to you a little bit about visualization using these same kinds of diffusion components, because I'll show you visualizations based on this throughout the talk. So we have a series of methods starting with FATE in 2019, multi-scale FATE in 2022, which visualizes this, to T-FATE, which has been useful in fMRI data. And we used a variant of this to visualize the worm data. So the idea of FATE is that we want to preserve the manifold geometry. So if you have data like this, if you sample it with noise, we'd like to see an embedding in 2D that looks similar. But if you look at different dimensionality reduction methods, they preserve different things, not necessarily the data geometry. TSNE and UMAP tend to preserve nearer neighborhoods. So they will give you this kind of structure, which could be useful if you're looking more for clusters. And then force-directed layout is trying to push everything away. Diffusion maps and isomaps. So isomap looks at geodesic distances on the graph, but if you have a noisy graph, it'll look a little bit noisy. But both of these have the disadvantage for visualization that they capture information in very high dimensions. So roughly speaking, each trajectory is encoded by 1 diffusion dimension. If you have 20 trajectories, you need 20 dimensions to look at it. But instead, we wanted to look at it in 2 or 3, and that's where FATE comes in. So the FATE method that we have, again, we compute these T-step diffusion probabilities, and then the next step we do is we log transform these. This gives you what we call a diffusion potential, and it allows us to compute an information theoretic divergence. You could use KL divergence, but we want it to be a distance, so we used a symmetric divergence. Then finally, we use MDS to embed this. MDS preserves these new distances as well as possible, so it gives you an embedding that's actually quite different than a diffusion map embedding. Here it's shown on single cell data of human embryonic stem cell development. And you see PCA here gives you the global structure, but it can't denoise in these nonlinear directions. TSNE gives you the blobs, and you see this is the developmental ordering projected on top based on the time at which these samples were collected in this 27-day time course, and you see the time structure is not preserved in TSNE. Diffusion maps do contain the structure information, but they contain it in high dimensions, and when you project it to low dimensions, you get kind of a projection effect, and that's FATE. We've used FATE on neuroscience data by using a multi-scale kernel, where you have each time point of neuronal activity high-dimensionally on the brain, and we use 2 views, a temporal affinity and a affinity between different time points and an autocorrelation kind of affinity. And when you combine these 2 views together, you can reapply FATE and it'll visualize trajectories. And you see that if you have toy data, again, you can see this toy data was just data that's kind of linear, like it's kind of following in a one-dimensional trend. And you see that some of the methods don't preserve it. If you think FATE is good enough, it's really not, because neuroscience data has even more noise than regular data, and we need the autocorrelation kernel that kills noise temporally as well as in the state space. So in this study of connectomics, we replaced this autocorrelation view with an inter-slice connectivity and intra-slice connectivity. Actually, these are flipped. Intra-slice connectivity means how are the neurons connected within a slice, and inter-slice is between iterations of the diffusion condensation. And this gives us the C-fate view, which is like a visual rendering of the diffusion condensation process. So you see everything going into one, and you see that, 4 branches here and sub-branches, and initially everything starts out as a single neuron. So it's a simple visualization of a fairly complex process.
So you just say the bottom one is inter.
Yeah.
So does that mean everything is basically
So it's like
diagonally
you have
coupled?
neurons. You have a neuron, and you connected it to itself in the next iteration of the condensation.
Everything is connected to itself?
Yeah. And so we've used this on cellular analysis, so I'm trying to show an example. This is actually from a neuronal organoid, which is a retinal. So this is fate. It gives you the different branches. Diffusion condensation gives you different clusters at different levels of granularity. The most persistent clusters actually turn out to be well-recognized ones, but it seems like the interesting science in this system was in a subcluster that arose much earlier in the condensation process, where you have these activated astrocyte states with a very specific signature for these states. This signature we were able to find actually in other diseases. So we found it in age-related macular degeneration, we found it in Alzheimer's and multiple sclerosis, which indicates that it's picking up on some kind of structure even in its earlier iterations pretty faithfully. So now let's go back to the connectomics data. This was again the NeuroPill or nerve ring data set. It has 181 neurons, but there were over 100,000 instances of neurite to neurite contacts, which is what we were given as data directly from electron microscopy images. And we did ask our collaborator why use these contacts rather than some kind of synapse information. And they told us they wanted to focus both on structural and functional properties of this, which would be easier to get from the contacts. So what we did was, so basically this is what the kind of data looks like. It actually is a segmentation from a fairly famous earlier paper by White. And it has these salami slices of the worm, and you can segment these in this way. And once you segment these contacts between neurons, you see the different contacts between different neurons are colored, form the adjacencies for us. So how do we go from these contacts to adjacencies? We use this contact fraction measurement where SA is the surface area. And this is sort of a Jaccard type distance. It's looking at the area in which the two neurons make contact, and it's normalized by the surface area of the entire neuron. And we'll show you a little bit later why we do it this way instead of just summing it up or something like that. So now we have a neuron by neuron adjacency matrix. Now, in order to go from adjacencies back to coordinates to run the diffusion condensation method, we do just use MDS for starters. And then the diffusion process takes over and computes diffusion condensation iterations. This is the whole condensation iteration process shown as a Sankey diagram, and I'll show it in a second as a C-fate diagram. So initially, everything starts out as a single neuron, and you get larger and larger groupings of neurons. These colors indicate known members of a mechanosensory circuit, for which we added the yellow neurons, which are actually very closely connected. One interesting thing about this process is if you visualize it like this, the length of the branches is very interesting because it indicates persistence. So it's not like hierarchical clustering that's forcing a cluster. It's actually going to tell you how long it stayed by itself. Okay, so then we use some analysis of different levels to figure out which levels that the neuroscientists wanted to study further. And we ended up using modularity, which is a measure of cluster separateness. And the most modular cluster level is when there are 4 clusters. So they did a lot of analysis on this. So you see these 4 clusters colored on the C-fate diagram that we've rendered in 3D. And these are called strata in the paper. And actually, these are the neurons that are in the strata. And then there are these unassigned neurons. And these unassigned neurons, even at this level, were separate. So they haven't formed a direct circuit with any of these. But it turns out a lot of these are members of what we call the rich club. They sort of connect with all these circuits. So they're forming some kind of crosstalk between all these circuits. So what is the significance of these 4 strata? It turns out that there is some anatomical and structural significance to these. The 4 strata or layers stack along the anterior posterior axis of the animal. And when you just look at them in images, they look really tightly bundled. So you might think there's no structural separation, but the computational method indicates that. So our collaborators did further study of this. So one of the things that's pretty interesting is strata 1, which is shown in red, sort of encases the other strata like an envelope. And the unassigned ones are sort of on the border of this. And then strata 2, 3, and 4 are inside. So it's kind of like hugging 2, 3, and 4. So then they looked at the axons that are leading from basically the mouth of the worm. And there's 2 kinds of axons, papillary axons and amphidial axons. And they saw that these separate. Strata 1 gets the projections of the papillary axons. Strata 2 has no axons projecting, and strata 3 and 4 have amphidial axons. So there is some separation of sensory information between these 2. So there's also sort of a functional separation. If you look deeper into the S1 strata, there is a mechanosensory circuit that controls head withdrawal. And so there is kind of an ordering of the neurons themselves. They go from sensory to inner neurons to motor neurons. And these are the additional members of the cluster that we added based on what it looks like from the condensation tree that's closely related. And interestingly, this I actually didn't expect, but they did 4-dimensional in vivo imaging of these. And this is the worm during development, just hours after it's born. And it turns out the initial positioning, but not the final positioning of the neurons, follows the strata order roughly. So the S1 migrate to the anterior part of the embryo head, and then 2, 3, and 4 migrate to the posterior part. So this was, so in a sense, you're kind of looking at all of these neurons and thinking of them as architectural components that aren't necessarily completely functional, but they're also structural, kind of in a way that computer architecture is. So more recently, and this is unpublished, our collaborators started asking the question, how much of the development of the worm from then on, from L1 to L2 to L3, is just scaling up? Meaning, do the neurons just get bigger and gain more and more surface area? And so this is because there's this data that we found in this Vliet paper, which I think Elad also showed yesterday. And you can download the adjacencies and connectivities of worms at all these different levels. So we sought again to use TDA to study allometry here. So diffusion condensation has a first-order homology based on the clusters that emerge. But by using the contact area fraction, it turns out that these persistence diagrams are invariant to allometric scaling. And that's because if you just had surface area that's twice as large, it would just cancel out here. And so the scaling factor doesn't really matter. So then what we have is these persistence diagrams from the condensation that are one-dimensional like this. All the neurons are individual at the time of birth, and then they merge, and each merge event shows the death of some of the components. And you see these components are eventually merged to a single point, which I showed you the rough proof for. And once this happens, you can start to compare different stages of the worms. And in order to do this, we use Wasserstein distance. So we actually use this 2 Wasserstein distance. I know some of you have heard of 1 Wasserstein, which is called Earth Movers distance. So you can do a distance matrix of Wasserstein distance between the worms now. So this is a way of getting distances between the entire connectivity persistence diagrams. So what we see here is that there is quite a bit of similarity. The nerve ring does have allometric scaling. At similar ages, the relationships are more similar. And the level of allometry increases as the worm matures. So what you can see here is between L1 at 0 hours and 5 hours, it's very similar. So that's a lot of allometric scaling. But then there's a jump here where there's a difference, so something reorganized. And then between L2 and L3, there's more sort of scaling, and then you see these jumps in reorganization that happens. So if you look at the C-fate diagrams of these, they, to a first order look pretty different. One thing is that 4-strata modularity is present since early development, and they, map to the same anterior-posterior regions. But it seems like really where there's variabilities in the level of persistence across these regions. So in some regions, there's more persistence. The most persistent region is the strata 1, which I keep visualizing in red. And we sought to ask, why is that the most persistent region? And actually, here's more proof that it's the most persistent. This is showing Wasserstein distance between this and others as time goes on, and you see that the red has the least Wasserstein distance corresponding to the most persistent, and S2 shows the greatest allometric relationship changes. And it turns out structurally S1 is this kind of flat.
Can I ask a quick question? Can you go back to that last slide?
Yeah.
Is there an explanation that you know of for the big dip for L3?
That I don't know, actually. I don't know if it's known yet, because we're still trying to explore this.
Okay.
Sorry. To compute the Wasserstein distance, you need to convert things to probability distributions,
Yeah.
right? Can you say again how you do that?
So you can actually form a density estimate just using this persistence diagram. You can form a one-dimensional density estimate, and then you get a Wasserstein distance.
You just put like little
Yeah.
Gaussian kernel and stuff.
Exactly. Sorry. So again, this is just work that's under development. It's not. So there's also a lot I don't know about the second order homology, which is a really good question that I want them to compute. So one thing we know about S1 and why it might be more persistent is it's sort of forming a platform on which the others are scaffolding. And so this seems to be the most conserved across the worms and between the developmental stages. Okay, so the summary of this sort of part of this story is that these EM-derived adjacencies can be used to study the whole organization, so not just a couple of clusters, but different kinds of organization. And the diffusion condensation that we applied to it, you can think about it as a kind of a TDA-like filtration that happens on a data manifold. And this method did help our neuroscientist collaborators delineate circuitry. And more recently, we've seen that TDA, because of its invariances, can be used to study allometry between developmental stages. But now I'm going to switch a little bit to go from structural to functional connectivity. So functional connectivity is often of interest because it's thought to be, so structural connectivity can be kind of all the roads in Los Angeles, but functional connectivity can be what roads people are all actually taking. And to me, it seems like 405 is the road that everybody's always taking. So it's always crowded. So that kind of thing. So one idea we have for going from structural to functional connectivity is to use a graph neural network where we start with a connectome. We place some key features on here, whatever we have. And if we don't have many features, then it can just be a prediction task. Then what we're training the graph neural network to do is to predict the dynamics of some kind of activity. It can be calcium signaling or whatever kind of activity measurement you have. And what we do is we use a form of graph attention to actually read out what the graph looks like. This was actually recently published in December 2023 in actually a new machine learning conference called Learning on Graphs. So the way it works is you use dynamic activity as the prediction task, and you have a graph, and the graph is a graph ODE, or a graph ordinary differential equation. So it's trying to learn the dynamics of the system given the input within itself. And so there is a small neural network, or MLP, at each of these nodes that integrates input from its attention neighbors and decides what the next value would be. And so we train it on these dynamic traces that in neuroscience we have, but in cellular and molecular stuff, we actually have to infer. And at the end of the day, you get these attention readouts, and you can process them for confidence levels to get a final sort of time-varying graph from this. So this is kind of interesting because you can take a static average of this to see what the average functional connectivity is. But one thing about the functional connectivity, like the road system, is the connections that are emphasized can change with time depending on what you're doing. And so actually we get a dynamic functional connectivity graph here instead of a static one. Yeah.
What are your free parameters for the training? Are they all associated with connections in the graph, or are they also associated with the dynamics on the node?
They are associated with the dynamics on the node. So we're learning a general MLP that governs the dynamics on the nodes. And one of the things we've been kind of interested in is, can we add more structure there? Are there particular kinds of dynamics we want to incorporate? But for starters, we assume we don't have a lot of knowledge on these systems and leave it free.
So the dynamics, can you say again what the dynamics are? GL?
I can show you the dynamics.
So you said there's an attention layer at every node?
I'll show you.
Okay.
Okay, so we actually put in a new kind of attention mechanism here. This attention mechanism, we call it space-time attention. Really what it is attention over neighbors as well as itself. And what the autocorrelation attention is also over its own time traces. And this is done, over a period of time before you compute the derivative. And so you can think of the graph ODE network as a network that predicts the derivative. Most neural networks predict a function or predict the output of the function. This network predicts the derivative. And so in specific, to get the function, you do have to integrate it out with a neural ODE, and then you're penalized by the output of the ODE. And of course, neural ODEs came out in 2018, and the really clever trick is that you don't really have to backpropagate through the ODE solver. You can use this adjoint sensitivity method from meteorology to bypass ODE solving. So this kind of attention, gives the time-varying graph, these attention reads. It supports hysteresis or integrate and fire type things because you can accumulate signal, and it does output an instantaneous derivative. So the actual ODE looks like this. It's a function on top of this derivative network, and it's the actual function, if you want it, is put into the ODE solver. I hope that answered your question about what the dynamic is.
So the loss function depends on just the steady state of these dynamics, or it depends on the entire trajectory?
You use every time point to penalize. That's how you build up enough data to train these. So this F is the MLP that gives you the dynamic.
So as these dynamics unfold, you want it to reproduce the recorded neural data.
Exactly, yeah. So the actual loss function looks like this.
Does it reproduce or predict forward?
It predicts forward. So you have the mean squared error as the loss function, as you just said. We also have closeness to a prior, which is useful in some cases, as well as a sparsity penalty. And so this is kind of the loss function that's inside here, yeah.
And so the thing where you said like every node has attention over time or something.
Yeah, it has attention over a period of time.
So so it's not its entire past history that it can
Not the
access.
entire past history, yeah. And that's actually kind of within some reason learned between 0 and tau. It's bounded by tau.
So can I think of this as like a really complicated version of like fitting a linear dynamical system to some
Not
time series
linear.
data? Yeah, hence the really complicated version.
Oh, okay. Okay, sure.
That's a generalizing a model, right? Where
Yeah.
node I looks at node J with a filter that has at most time tau.
Right,
into the
that's
path.
right, that's right.
It's like a VLM.
Yeah.
sorry, maybe this is too detailed for your talk plan, but like the top line there is G prime equals F theta of G prime, comma T, right? So there's no ODE anymore. It's just a nonlinear relationship that you use to update the derivative of G.
So the neural network embodies so you can time query the neural network. You can give it a time, and you can give it an input, and it'll give you the instantaneous time derivative. So that's how it works.
Or why does the F theta depend on G prime and not G?
Oh, I think that's a typo. Yeah. Sorry about that. So one of the interesting things here is that you could apply a perturbation and look at its effects. And actually, if you had such a perturbation, you could also train with a perturbation. So that's something that we're still looking to get data for. So what did we use for this to validate this? We used a couple of simulations. So I used a cellular level simulation. I just want to make sure I'm finishing on time. A cellular level simulation of cellular dynamics. So you have a gene regulatory network within a cell. And based on the activities of the gene regulatory network, the cell changes its transcriptional program and could even differentiate into different types of cells. So we modeled that. And then in the cellular landscape, you actually don't have time-varying measurements. So you can use something called pseudotime. And to get individual cellular trajectories, we have another framework called MioFlow that I won't show you. So our neural network is trained to match the trajectories of MioFlow, whether it's this trajectory or this trajectory. And the idea is to re-infer the underlying gene regulatory network. And in specific, you see that the connections that are active at different parts of the state are different in a way that we see it. And we can look at it and compare it to other methods, most of which are static. So we actually have to take the time average of our dynamic graph to actually make this comparison. So we've also tested it on sort of realistic data of human embryonic stem cells differentiating into neural progenitor and neural crest. So you see the neural progenitor can be segmented as the top branch, and the neural crest is segmented as sort of the bottomish branch. And we use something for trajectory inference that does dynamic optimal transport. Actually, we love optimal transport in our lab. If you're interested, you can look at that paper. And then we have an inferred gene regulatory network that goes from early to late in the system. So for neuronal simulators, we actually looked at the nest simulation. These are these coupled oscillators that have these coupling equations, one for excitatory neurons and one for inhibitory neurons. Probably a lot of you are more familiar with this simulation than I am. And so to mimic optogenetic stimulations, we usually altered this resting membrane potential type parameter, and we did the same thing. But here you see that our method doesn't always outperform these other methods. The reason is because this is just a static graph, and it's actually fairly simple. We're hoping that when we get into more complicated neuronal systems, that the RITINI will more consistently outperform these methods. So the final thing that I wanted to show you is that once you have this kind of a graph, you can really do a lot of things. You can think of any kind of neuronal activity as cool patterns that are playing out on this graph. And we have a bunch of work that does that. So you have some kind of a graph, there's activity. We use, again, these graph signal processing type techniques to map out the activity. We can get time traces of the activity and then persistence diagrams that quantify them. I won't get into the details of the signal processing. But for example, when you do this visually and persistence diagram-wise, you can see large differences between neuronal and epithelial layers, as well as differences between unperturbed dynamics and perturbed dynamics. So that's something that we do post hoc, and I just wanted to show you that because I talked about just getting the graph the whole time. And if you're interested in any of the tools, they're usually on my GitHub.